This is our cell signaling project video by Lindsay Zhang and Natalie Sun in period 3. Ozempic is an anti-diabetic medication and weight loss drug that has been popularized for its use among celebrities such as Oprah Winfrey, Kelly Clarkson, and Amy Schumer. Ozempic's generic name is semaglutide. Semaglutide is a glucagon-like peptide receptor agonist, meaning that it mimics human-made GLP-1 and activates the GLP-1 receptor. Semaglutide is a hydrophilic molecule because GLP-1 receptors are transmembrane proteins that span the surface of beta cells. The semaglutide pathway involves G-protein coupled receptors called glucagon-like peptide 1 receptors. GLP-1 receptor proteins are typically used to control blood sugar levels by increasing insulin production and reducing glucagon secretion in the cell, making its agonists useful for treating type 2 diabetes as they lower blood sugar. These receptors also increase feelings of satiety, slow down gastric emptying, and improve glucose disposal in surrounding tissues. Some side effects that can occur from using Ozempic or semaglutide include nausea, headache, and heart palpitations.
The ligand involved in the GLP-1 signal pathway is semaglutide. Its receptor is a GLP-1 receptor. When semaglutide binds to a GLP-1 receptor, it causes a conformational change in the receptor. This activates the GS protein, which exchanges GDP for GTP and activates its GS alpha subunit. The activated GS alpha subunit dissociates from the beta and gamma subunits and binds to adenylyl cyclase, catalyzing the conversion of ATP to cyclic AMP enzymes. The rise in cyclic AMP enzymes activates protein kinase A. pKa closes the ATP-sensitive potassium ion channels in the cell membrane. This prevents potassium ions from flowing out of the cell, building up positive charges and causing cell depolarization. Cell depolarization opens the calcium ion channel, increasing their concentration in the cell. The calcium concentration stimulates insulin exocytosis by fusing with vesicles containing insulin, which then leave the cell. GLP-1 receptors also inhibit glucagon release, which helps keep glucose levels down. The termination of the semaglutide signaling pathway involves several key mechanisms. After semaglutide binds to the GLP-1 receptor and activates it, the receptor can undergo phosphorylation by G-protein-coupled receptor kinases. This causes further interaction between the receptor and G proteins to be blocked and stops additional signaling. The GS alpha subunit hydrolyzes GTP to GDP over time. Once it becomes inactive, it can no longer activate adenylyl cyclase, thus halting cyclic AMP production and effectively stopping the primary signaling cascade initiated by semaglutide. Cyclic AMP is also regulated by the phosphodiesterase enzymes, which break down cyclic AMP into AMP, reducing cyclic AMP levels in the cells and terminating pKa activity. Semaglutide is slowly metabolized in the body by proteolytic enzymes and cleared by the kidneys and liver. As the concentration of semaglutide decreases in circulation, fewer GLP-1 receptors are activated, reducing signaling over time. The primary intended response of the cell is to increase insulin secretion in pancreatic beta cells. GLP-1 receptors in the brain promote feelings of satiety, which can help manage appetite. GLP-1 receptors in the stomach slow down gastric emptying, leaving you feel fuller for longer. All three of these cell reactions help with weight loss by reducing appetite and type 2 diabetes by regulating blood sugar levels.
Thank you for watching our cell signaling project on Ozempic.